Hello, my name is Catia Pierotti, and I'm a postdoctoral fellow at the Walter and Eliza Hall Institute in Melbourne, Australia, in the labs of Professor Guillaume Lessene and Professor James Murphy. My research interests are centered around investigating the mechanism of action of small molecules that inhibit necroptosis. Necroptosis is a form of programmed cell death. Unlike apoptosis, which is the ordered dismantling of the cell executed by the caspase family of proteins, necroptosis is a lytic inflammatory form of cell death, characterized by loss of plasma membrane integrity and the spillage of the intracellular contents. Necroptosis is involved in the pathophysiology of a wide variety of human diseases, including inflammatory diseases, degenerative conditions, infectious diseases, and cancers. The best characterized form of necroptosis is mediated by the TNF signaling pathway, where TNF binding to its receptor initiates the signaling cascade, which ultimately results in the formation of a multi-protein complex containing the key protein effectors RIP1, RIP3, and MLKL, which activates MLKL to cause membrane permeabilization and cell death. In an experimental setting, necroptosis can be induced by treating cells with the cocktail TSQ. So the three key proteins that regulate necroptosis are the kinases RIP1 and RIP3 and the pseudokinase MLKL. In previous work performed at WEHI, we identified a small molecule called compound 1 that binds to murine MLKL and inhibits necroptosis in murine cells. In this study, we investigated a more potent analog called compound 2, and we sought to determine its protein targets and mechanism of action. We first determined if the compounds bind to the necroptotic effectors in vitro, and for this we used competition binding assays from the DiscoverX Kinome Scan commercial platform and obtained the binding affinities, or KD, listed. This demonstrated that compounds 1 and 2 do bind to MLKL, RIP1, and RIP3 in the nanomolar to micromolar range. We next wanted to determine if compound 2 binds to MLKL, RIP1, and RIP3 in cells. We used CETA to perform cellular target engagement, where compound binding confers thermal stabilization or destabilization of the target protein as monitored by Western blot. In human U937 cells, compound 2 decreased the thermal stability of MLKL, increased the thermal stability of RIP1, and slightly decreased the thermal stability of RIP3, altogether demonstrating that compound 2 binds to MLKL, RIP1, and RIP3 in human cells. We then determined the ability of these compounds to inhibit necroptosis in cells. Human U937 cells were treated with TSQ to induce necroptosis, and cell death was measured by propydium iodide uptake and analyzed by flow cytometry. Compound 1 inhibited necroptosis at nanomolar concentrations, while compound 2 was more potent and inhibited necroptosis at low nanomolar concentrations. We then wanted to evaluate the ability of compound 2 to block necroptosis in vivo. We chose the TNF-induced SIRS model, where injection of TNF causes a systemic inflammatory response and lethal hypothermia in mice. And this can be monitored by a decline in core body temperature of the mice. In this model, the compound 2 treated mice displayed a delayed decrease in core body temperature and a modest increase in overall survival. This demonstrated that compound 2 shows efficacy in vivo and provides protection in this model. In conclusion, compound 2 targets all three necroptotic effectors, MLKL, RIP1, and RIP3, to potently block necroptosis. I would like to thank and gratefully acknowledge my supervisors, colleagues and collaborators, funding sources, facilities, and affiliations who contributed to this work. Thank you.